Welcome to Module 5 of Cells, Tissues and Regulation. In Topic 5.2, we will learn about endocrinology. Topic 5.2 itself is divided into two parts, 5.2a and b. In 5.2a, we will learn about chemical signals, the classification of hormones, and their mechanism of action. During the course of this video, you can open your workbooks to Module 5 and try and answer as many questions as you can from Topic 5.2. The learning objectives for this topic are to describe autocrines and paracrines and their differences, to describe ligands, agonists, and antagonists, to define hormones and the types of cells that respond to hormones, to distinguish between amino acid and steroid hormones and briefly outline the role of eicosanoids, to explain when hormones bind to receptors, the different types of intracellular events that occur to produce physiological responses, to also explain when water-soluble hormone binds their receptors, and describe the chain of events that occur that can lead to an intracellular response in the target cell. And finally, in regards to steroid hormones, describe the sequence of events leading from the time the hormone arrives at the cell surface to the synthesis of a new protein by the cell, and why the process of protein synthesis is important. As discussed in the previous video, the two major communicating systems in the body are the nervous system, which regulates the activity of muscles and glands via electrochemical impulses that are delivered by neurons. These signals are very rapid and precise, and the organs respond within milliseconds. And the second, which is the endocrine system. The means of control and the speed of the endocrine system are very different in that they influence the metabolic activity using hormonal signals. Hormones are chemical messengers secreted by cells into extracellular fluid. These messages travel through the blood and regulate the metabolic function of other cells. In the body, binding of hormones to cellular receptors initiates responses that typically occur over a period of a few seconds or even days. Once initiated, these responses tend to last much longer than those created by the nervous system. The endocrine system controls several important functions in the body, such as the regulation of water and electrolytes, inducing stress reactions, controlling our growth, reproduction, and so on. However, the endocrine system and the nervous system do coordinate and integrate several critical functions in the body, including circulation, digestion, metabolism, and mobilization of defenses. We have briefly discussed the differences between the endocrine and exocrine glands in Module 1, Topic 1.3. Just to review, endocrine glands produce hormones and lack ducts. They release hormones into the extracellular fluid. Several major endocrine glands are widely scattered throughout the body. Although small, these organs can have a powerful influence on the body. These glands have a predominant endocrine role, meaning they excrete hormones. However, some of these may also have major exocrine functions. For example, the pancreas secretes several digestive enzymes into the gastrointestinal tract, therefore working as both an endocrine and exocrine gland. Furthermore, there are several other organs which, even though they secrete hormones, are not classified as endocrine glands because their primary role as an organ may be different. For example, the kidneys produce several important hormones that are essential to our survival, such as erythropoietin, that controls the production of red blood cells. The chemicals, or hormones, are classified into three different types. Autocrines are chemicals that exert their effects on the same cells that secrete them. For example, growth factors to promote proliferation of the same cell type. Another example are cytokines and interleukins that are actively secreted by white blood cells during an immune response. These autocrine chemicals allow the white blood cells to multiply rapidly and fight the immunogenic material. Paracrines are locally acting chemicals within the same tissue. These chemicals affect the cell types other than those releasing the paracrine chemicals. The diffusion of paracrines is limited by distance, and these chemicals are rapidly inactivated. Some examples for paracrine chemicals include histamine, a compound that is involved in local immune responses, prostaglandins, which have a wide range of functions, nitric oxide, which is an important vasodilator, growth factors, and blood coagulation factors. Endocrine chemicals are long-distance chemicals that travel in the blood throughout the body. These include the hormones secreted by the pituitary, thyroid, and adrenal glands. Ligands are any molecule, such as hormones, growth factors, or neurotransmitters, which can bind to cell receptors. Once bound, the ligand can activate or deactivate a cell or its functions. These ligands can act as either agonists or antagonists. When a ligand binds to a receptor and activates a physiological response from that receptor, it is called an agonist. When a ligand binds to a receptor and inhibits the response from the receptor, it is called an antagonist. Some molecules in small doses may act as agonists. However, when a tissue is bombarded with the same molecule in large doses, it can overload the system and have an antagonistic effect. Hormones, by definition, are chemical substances secreted into the blood in very small quantities by specialized cells or glands, and carried by the bloodstream to other parts of the body, where they interact with specific receptors on the target cell to produce a particular response. The chemical structure of the hormone determines its water solubility, which is one of the most critical properties of a hormone. The water solubility of the hormone in turn affects how the hormone is transported into the blood, how long it lasts for before it is degraded, and what kind of receptor it can act on. Although a large variety of hormones are produced, nearly all of them are chemically classified into three main groups: amino acid-based hormones, steroids, and eicosanoids. Most hormones that are produced in the body are amino acid-based. These hormones are synthesized and can be stored in glands. Because amino acid-based hormones are water-soluble, they cannot cross the plasma membrane. The molecular size varies widely in this group, from simple amino acid derivatives that include biogenic amines such as thyroxine, peptides that are short chains of amino acids, such as insulin, and proteins that are long polymers of amino acids, greater than 50 amino acids, for example, such as follicle-stimulating hormone. Steroid hormones are synthesized from cholesterol. These hormones are very similar in structure and are released only from the gonads and adrenal cortex. These hormones are lipid-soluble and can therefore cross the plasma membrane to interact. Eicosanoids are the third class of hormones. This group includes leukotrienes and prostaglandins. These are biologically active lipids that are highly localized, affecting only nearby cells. Eicosanoids act as paracrine and autocrine chemicals and have a hormone-like activity. Leukotrienes are signaling chemicals that mediate inflammation and some allergic reactions. Prostaglandins have multiple targets and have effects ranging from the control of blood pressure to inflammation, pain, fever, clotting, renal function, protection of the gastric mucosa, and smooth muscle contraction. Aspirin, which is one of the most commonly used anti-inflammatory drug, reduces swelling and pain of a sprained ankle, for example, by inhibiting the release of prostaglandins. As mentioned previously, the chemical structure and the water solubility of a hormone determines how it is transported in the blood, its duration of action, and what receptor it can bind to. Peptide and amine hormones are hydrophilic, meaning water-soluble. Because these hormones cannot diffuse through the plasma membrane, they need cell surface receptors that signal into the cell by a secondary messenger system. These hormones have a rapid onset and a very short duration of effect. On the other hand, steroids are hydrophobic, meaning they're lipid-soluble. Therefore, these hormones can diffuse through the plasma membrane and use intracellular receptors to bind within the DNA and alter the gene expression. Steroids have a slow onset and a long duration of effect. Thyroxine is the only hormone which, even though it is composed of amino acids, can move through the cell membrane to bind to an internal receptor, just like steroid hormones do. With the exception of thyroxine, all of the amino acid-based hormones exert their signaling effects through intracellular messages generated when a hormone binds to a receptor in the plasma membrane. Whenever a hormone binds to its receptor on the plasma membrane, it causes the receptor to change its shape and bind with a nearby inactive G protein. Once bound, the G protein gets activated and is displaced from its position to bind with an effector enzyme, adenylate cyclase, which produces second messengers, such as cyclic AMP, that trigger the activation of protein kinases. These protein kinases then trigger responses of the target cell. This might be activating enzymes, stimulating cellular secretions, or opening ion channels. Steroids and thyroid hormone, being lipid-soluble, can diffuse into their target cells, where they bind to and activate an intracellular receptor. The activated receptor hormone complex then makes its way to the nuclear chromatin and binds to a particular region of DNA. The receptor hormone complex binds to the DNA and initiates the transcription of DNA to produce messenger RNA. The messenger RNA is then translated by the cytoplasmic ribosomes, producing specific proteins. The cellular effects of steroid hormones develop more slowly than the second messenger systems, such as the amino acid base hormones. However, many steroid hormones also have cell surface receptors that use a similar second messenger system for more rapid cellular responses. In summary, from this video, we have learnt an overview of two major communicating systems in the body, the difference between autocrine, paracrine, and endocrine signaling, ligands and their role as agonists and antagonists, hormones and their classification, the action of water-soluble hormones, and the action of lipid-soluble steroid and thyroid hormone.